Hello everyone, this is Suman. Welcome to another video from DNA Bioscience. So, in the last class, we have discussed about introduction of VTC recombination. We have discussed the VTC recombination in case of variable regions, both in the light chain and heavy chain. And the difference is the light chain, it lacks the D chain, right? So today we are going to discuss about the recombination in the constant region, about the 12 by 23 rule, and about the mechanism of VTJ recombination. So let's begin with the arrangement of the constant region. So in the constant region, as I have marked, 5 genes are present for 5 individual immunoglobulins. The circle I have marked for the potential recombination sites. What does it mean? That means the recombination should occur in this particular area. So, like the rearrangement in the variable region, the constant region is not that much of active. The variable region, the VDJ, the genes are shuffled and they are rearranged with each other, deleted and form the arrangement which produce a new expression. That's why they are called hypermutate, and it is a kind of deletion mutation. As we have discussed, to make an arrangement we have deleted all the sequences that are present in between them. So it is a kind of deleted mutation, and it is hypermutation in case of variable region, and as it is occurs in the somatic cell, that's why it is called somatic hypermutation. If you delete this part, then the immunoglobulins are G, E and A. Then if you add the variable domain with the constant chain, that is the V, D, J with G, right? So the immunoglobulin here is IG G type. So the constant region arrangement, it says about what kind of antibody is going to be produced, correct? If the B cell first recognizes the antigen and forms the IgM immunoglobulin, then the B cell can make any of the other immunoglobulin. But if the B cell make the IgA type, then the B cell cannot make any other preceding immunoglobulins. Why? As in moving forward, the B cell discard and delete all the preceding immunoglobulins. That's why the name is deletion mutation. If recombination not occur and the B cell fails to recognize the pathogen, then by default IgM immunoglobulin will produce. So this is the idea. So let's switch to the mechanism. The RSS, that is called the recombination signal sequence, it is bind to all the genes, that is the variable genes. Here in this case we are dealing with the V and J genes. So the RSS sequence are present. Then in the next step, the protein complex that is called the RAG1 and RAG2, it binds to the recombination signal sequence, correct? So what is the first sequence? First the recombination signal sequence binds to the genes, and then the RAG1 and RAG2 protein complex, it binds to the RSS. Okay, then the next step is the formation of synapses. Synapses, we know in crossing over we have studied about the synapses. So synapses is the pairing of the homologous chromosomes. So what the RAG1 and RAG2 will do, it creates a single stranded nick in the DNA. These are the nicks. So it creates the single stranded nick in the DNA between the first base of the RSS and the coding segment, which results in the formation of 3-OH group. Then the reactive hydroxyl group is positioned by the recombinase enzyme to attack the phosphodiester bond of opposite strand, okay, forming two DNA ends, that is the hairpins on the coding segment and a blunt end on the signal segment. These are the signal segments, the blunt ends, and in the coding part, two hairpin structures. Then the coding ends are further processed by extra some other protein. So the fourth step is binding of DNA, protein kinases, and hairpin opening. Okay. So the additional proteins, that is the DNA dependent protein kinase and a dimer of DNA ligase and XRCC4 are incorporated into a large complex with the RAG proteins. Correct. So this is the RAG protein. Some additional proteins are joined. These RSS ends are joints forming what is called the signal joint. So here the process becomes DNA ends occur to create a closed circular DNA that plays no further role in the recombination process. So the DNA hairpins at the ends of the gene segments, they are cleaved. So in this case the hairpin is open and cleaved, and in the next step, that is called the processing of DNA ends, the segments hairpin at the ends of the gene segments after cleavage, an additional enzyme that is called DDTT, that is called deoxynucleotide transferase, is recruited and adds additional nucleotides at the ends of the DNA strands. The other enzymes in the complex like it together. So what the DDT does, the DDT, it provides additional nucleotides to the ends of the DNA strands. So you can see these are the additional nucleotides. The further enzymes in the complex like it together the two ends of the gene segments, completing the recombination process. So the RSS segment, it is divisible into three segments. The first one is a heptamer. You can see in this picture, this is the V region, this is the D region, and this is the J region. So in the D region, you can see a heptamer of 7 conserved nucleotides are present in the V, D, and J. A spacer region of 12 or 23 base pairs in length. Gene segments to be recombined are usually adjacent to RSS of different spacer length. One has a 12 RSS and one has a 23 RSS. So here is the 12 RSS, here are 23 RSS. A monomer of 9 conserved nucleotides is also present. Here the monomers of 9 conserved nucleotides. So the monomer is separated from the heptamer by 12 or 23 nucleotide spatial, and this rule is called the 12 by 23 rule, as the gene segments, that is the VDJ, which are going to be recombined, are usually adjacent to RSS of different space length, that is the 12, 112 or 123. Now the 12 by 23 rule, I hope it will help you. Please subscribe to my channel to get more such videos, and if you like this video, please hit the like button. Thank you.